Thanks, Andrew. As Andrew said, you can kind of see from the list of co-authors that we have here, there's a very, very pertinent and very important issue that we have in Ireland that we put probably most of our focus on actually phenotypes from dairy cattle and from beef cattle and also sheep perspective. Just so we're all on the level playing field, just to start off with some terminology, phenomics, as we call it, is really just a discipline whereby we try to describe or characterize individual animals. And by characterization, I mean the phenotypes of the particular animal. And essentially, just phenotype is just a fancy word for trait. And that could mean milk yield, growth rate, fertility, or health, or whatever. And I think you can probably categorize phenotypes into around 7 different categories. And what I'm going to do in this talk is I'm going to go individually through these 7 categories, outline them, and talk about what are the advantages or the disadvantages of each of these particular categories. And usually when I start talking about phenotypes, people get all excited and they start talking about technical things and how you're going to take biopsies and you're going to do this and you're going to do that. But I honestly think what I've learned in the past few years is producer scored traits or phenotypes are actually probably the easiest and they're probably the most beneficial. And when you stand back from it and think about it, it actually makes total sense. Producers are all day long in with their animals, they know their animals the best, so they're going to be able to tell us as geneticists which are their good animals and which are their bad animals. The critics, of course, would say that every farmer's opinion differs on what's the best animal, and they're right. But if it was totally random, then the traits wouldn't be heritable. And I'll show you in the next slide that these traits are actually heritable, which means there is some degree of consistency in these particular traits across farmers. Also, I'm a farmer myself, and I know I've got great cattle, the best cattle in the world. I won't tell anybody I've got bad cattle. But really, at a genetic evaluation perspective, this doesn't really make a difference because it's all based on contemporary comparison. So as long as the farmers actually show variation, then we can use these in genetic evaluations and if the traits are heritable. And the analogy is that some farmers feed their cows differently. So we account for all that through contemporary group effects in our genetic evaluations. And of course, there's other people that say that just farmers won't score properly, they don't know how to score it, and that's not a scientific approach. But that's absolute rubbish. And this is just giving you an example of some of the producer score traits that we're scoring in Ireland. And Ross is going to give a lot more talk about this on the beef perspective, and that's what's really actually caught my eye on it, is the beef and the ability of farmers to consistently score high heritability traits that are very strongly genetically correlated with the end product traits, especially in beef. But if we just focus on dairying, farmer satisfaction or workability, it's scored in the likes of New Zealand, Ireland, and in Australia, and it's showing a heritability of around 12 to 23%. So there is good consistency there that we can identify particular family lines or bloodlines that farmers tend to like. And it is genetically correlated with survival. Calving difficulty, and there's a few posters on dystocia and docility in the next session. And the heritability for dystocia in Ireland is around 24%. And for dairy and beef cattle, the heritability of docility is around 7 to 36%. So farmers, again, are able to identify the docile animals versus the temperamental animals. The advantages, as you can gather, it's simple. Producers know their animals the best, so therefore they're going to actually be able to score them the most accurately. It is heritable, so there is consistency, and there is, and you'll particularly see this in Ross's talk, there is strong genetic correlations with the end product traits. And of course, there's a little marginal cost. The farmer is out there the whole time, he can have the notebook with him, and he can just start scoring those animals, and that's what we do in Ireland. The disadvantages, and when I would have started this kind of research, I would have said the heritability would be too low. There's inconsistency or subjectiveness associated with it, but as you can see from the previous slide, that's not true. These are heritable traits. But we do need an easy way to actually capture this data. And we're seeing the proliferation of the likes of smartphones. Every farmer almost has them now at this stage, so it's very easy to actually record these particular traits. And a case example would be the likes of mastitis and lameness. Farmer behind the cows, bringing them in. The lame cow is going to be at the back, or the mastitic cow is at the back, out with the phone, bang it in, straight onto the national database. Most people are familiar with the professional categories of phenotypes, such as linear type classification, veterinary events, AI services, hoof trimmers, and there's many, many more. The advantages, you would say, is that these are going to be better quality because they're being scored by trained professionals. But actually, if you look at the data where we have producer-scored information and professionally scored information, the professionally scored information is not any better than the producer-scored information at predicting the end product traits, especially for the likes of beef traits. And from a producer or from a professional perspective, there is very much a synergy between the national database of which houses all this data and drumming up business for the individual business plans. So, for example, veterinary services. The demand for the veterinary services can be improved by actually the vet having full access to all the data on the farm and being able to take more of a prophylactic look at the particular events. The disadvantage, of course, is the cost associated with actually training these professionals and getting them out on farms to score them. And I think it's about time that some good cost-benefit analysis was done on a lot of these traits, because now there are many alternatives. And one of the things came up this morning was on linear type classification, and what is the economic value of linear type classification when you have the true traits like mastitis, lameness, etc., scored potentially by the farmer in your national genetic evaluations. And of course, I'm going to talk about in a second the whole idea of automation or technological alternatives to some of these professionally scored traits, such as image analysis and infrared tomography. The third category is technological ones, and essentially what I mean by technological is just data that's automatically captured, and it mightn't currently be used in a phenotyping strategy, but by little tweaks, it could actually be very, very useful in both genetic evaluations, but also in day-to-day farm management. And when we talk about technological techniques, most people will automatically think of milk recording. But just an emerging field which is using these potential milk samples is the whole issue of mid-infrared spectroscopy of particular milk. So previously, when cows were milk recorded, you got back the fat, the protein, and the lactose concentration. But now there's plenty of studies outside there to say that we can actually use some the infrared technology to predict milk fatty acid content, lactoferrin content, the processability of the milk, energy balance, and even the methane emissions of the animal. And this is just giving you an example of what is this MIR or mid-infrared spectroscopy that we're talking about. Well, your cow is milk recorded, and the milk samples go into the milk analyzer in the milk lab. And what happens is that light is essentially shone in the infrared region through that particular milk sample, and based on the vibrations of the bonds that are inside in that milk sample, a spectrum is produced. This is in the mid-infrared region, so we call this the mid-infrared spectrum, or the MIR. This is already related to fat, protein, and lactose, but as I said, the studies, particularly those in robust milk, can show that this is related to actually milk fatty acid or lactoferrin and many other components. So the farmer can get a detailed report, other than just his milk, fat, protein, and lactose. He can get a detailed report of other milk or other animal characteristics or phenotypes. The advantages is really has a low running cost other than depreciation. There's no paper trail because it's all automatic or technological, and I have it as an advantage, but it could also be viewed as probably a disadvantage, is there's a huge amount of potential data that can come from this. And how do we actually package this, like what Dan was talking about, to the farmer to give them easy to use statistics that they can use? The disadvantage is possible a high capital cost. The example is the milk analyzers, but if they're already there, then there's no cost. But usually for a lot of these technological phenotypes, it does require a calibration, and this usually does incur a pretty high cost. We're halfway there, the fourth category of phenotypes I call statistical phenotypes, and most people here that are involved in breeding are familiar with BLUP, best linear unbiased prediction. That's the method of doing genetic evaluations, whereby we take a load of data and we adjust for the systematic environmental effects such as contemporary group, herd, parity, data calving, etc., and we extract out the genetic effects. As geneticists or breeders, we publish the genetic effects, but we tend to throw away the estimates of the fixed effects or the other random effects. But really, this information can be used by individual farmers to see how well is my herd performing, or even my first parity animals or my second parity animals, how well are they performing relative to their contemporaries after accounting for the superior genetics. And Dan actually touched on this one where he was talking about, as a farmer, I'm not feeding my animals as well as their genetic potential. But being able to actually extract this information from the national genetic evaluations will actually be able to provide that information to the farmer and be able to tell them more conclusively if he is a good manager or not. Does he have good fertility because he has good genetics for fertility, or is he just a good manager? The advantages, they're already there. They're byproducts from the genetic evaluations. We can improve our genetic evaluation models and maybe get more detailed phenotypes or more informative phenotypes, but at the same time, we'll also be improving our genetic evaluations. So it's a win-win. The disadvantage, you could argue, it's going to be difficult to explain because it might be relative to a mean of 0, it might be negative, it might be positive. But farmers are used to these type of figures from a blup evaluation perspective. Anyway, we can add something to the mean, or to the mean of the figures, but it actually, farmers are well used to these figures. It's just a matter of going out and explaining them. Stefan already talked about genomic selection, so I'm not going to talk about the contribution to breeding decisions that genomics can actually have. But Dan again touched on this whole feeding for different genes. And it's an emerging area in the human literature where we have this called personalized medicine. And an example is the BRCA1 and the BRCA2 genes, which are associated with cancer in humans. And if individuals are screened for these genes and they have the unfavorable variants or alleles, then there can be greater monitoring or there can be more prophylactic treatment of these individuals because we know they have a greater genetic susceptibility to these particular ailments. Cattle, exactly the same thing. We can use this type of information to actually help decide which are the animals that are more suitable to different systems. And we already do this. If you compare the North American or Canadian animals to the Irish or the New Zealand animals, you'll see they're very, very different. They've been selected on their holistic genotype profile for the different production systems. And examples that I'm talking about here are kind of greater susceptibility to disease. So if an animal is more susceptible, say, to mastitis, should we manage that animal differently? Should we actually use greater or a different dry cow product or mastitis product, or should we actually monitor the animal closely? If animals have greater growth rates, should we feed them differently? And we all do this in a feedlot system, is we pen animals that are a similar size or potential growth rate into different pens, and we might feed on the smaller animals. But really, we actually know that information from birth. We potentially can know that information from birth, so we can manage the animals very differently from birth. From a milk perspective, we can do product differentiation. And this is already done with A1, A2 beta-casein milk. It's just based on a tiny variant change in the beta-casein gene, and those cows produce different types of milk, which can go into different types of products. The main advantage of this type of approach is DNA is present from birth. It's the same throughout your life, and it's actually even present before birth, so even at an embryo level. The disadvantage, however, and this is usually underestimated, is it requires huge investment to actually be able to find the causative mutations or the functional mutations or the pieces of DNA that actually cause these effects in particular animals. And especially if we're talking about genotype by environment interactions, we need a huge amount of data to see how the expression of this gene differs depending on the different environments. And of course, animals are not as simple as we thought. But our understanding and the technology that's available to us to interrogate this type of information is improving year on year, so it's not insurmountable. The second last type of phenotype I want to talk about I call next generation phenotypes. We all are familiar with genes, but every cell in our body contains exactly the same genes, but different ones are being what we call transcribed into different organs. And that results in what we call the transcript. The gene expression or the messenger RNA then is then translated into individual proteins, and they come together to actually produce what we traditionally would have known as the phenotype or the particular trait. So we have genomics, moving on to transcriptomics, then proteomics and metabolomics, and then on to what I originally started off with, the term phenomics. But really, the whole lot of these traits or components could be thought of as phenotypes. And just to give you an example, going from the protein to the actual expression of the trait, we have a lot of farmer or recorder noise going in there. And an example is fertility, which we know is lowly heritable. But if we actually look at the hormonal profiles, like commencement to luteal activity based on progesterone, we know we can get up the heritability of the trait. And a lot of that is probably due to reducing farmer voluntary waiting periods, etc., or farmer recording. So potentially we can get greater heritability for these particular traits, albeit potentially at a larger cost in the current situation, but it could be greater heritability, resulting in greater genetic gain. So that's essentially the same thing. The advantages, it's more heritable potentially, and therefore greater genetic gain. And it's not just, you're not going to a sample and just measuring a particular thing like fertility. Because if you take it from particular biological samples, you can measure a holistic view of how is that animal performing in a number of different characteristics or phenotypes. The disadvantage at the moment is the cost, but it's not really a problem in the future. We're seeing the cost of sequencing is coming down considerably. So the technology is improving. This will be a phenotype of the future. Probably one of the more important ones is how do we actually procure the biological samples. So as I said, all of our cells have the same genes, but different ones are being expressed, be it in the legs or the udder or wherever. And it's how do we actually get a sample of that. But there is some work coming on in the likes of UC Davis that is looking at the transcriptome of milk samples as a predictor of the transcriptome of other biopsies. Final one is monitoring, and we see this terminology a lot, and I use it a lot. Breeding is cumulative and permanent, and we all think this is a good thing, but it could also be thought of as a bad thing. And a good example is fertility, and we had a few talks about this already, is why did fertility reduce in the Holstein breed? And it was because we weren't monitoring it. But as Brian showed, by actually putting emphasis on the fertility trait, by actually collecting the data, we can actually adjust genetic trends. And to answer Peter Amer's question, in Ireland for the past 3 years, we have seen a reduction in, or improvement in the genetic merit for fertility, and that has been followed by a phenotypic improvement as well. So much so that by 2020, we expect the fertility of the national dairy herd in Ireland to be at 1985 levels, even though we have a 50% increase in milk production in the same period. But one of the things, because I'm also involved in beef breeding, is I see this from a dairy perspective. But to me, around the world, the vast majority of this is also happening in beef cattle breeding. They're aggressively selecting for output traits, and we know that they're antagonistically correlated with fertility and, say, milk yield of the animals. And they're not recording those particular phenotypes. And I fear that it could end up with the same dilemma as the Holstein was, or the Holstein was 10 years ago. So from a monitoring perspective, what I would talk about is what is the impact of the current breeding goals on traits that are usually difficult to measure, so like health traits, disease traits, feed intake, environmental load, and welfare. But also, how will these animals perform in different environments? And in an EU perspective, especially in the likes of Ireland, post-quota, how will our bulls, our elite bulls of today, stack up versus the elite bulls of 2015 and greater? And you have to remember, where does the data for these genetic evaluations currently come from? It comes from a distribution of farms, the average distribution. But will the average be the average in 10 years' time, or will it be to the right of the distribution of these farmers that are actually the farmers of the future? And how are they managing their animals? So if you want to test this out, you've got two main options. One is you can just go large-scale phenotyping, estimate genetic parameters, do selection indexes, and derive the expected responses to selection. This can cost a lot of money, so another alternative is to do smaller, controlled experiments where you actually have divergent selection lines. One, which would be genetically elite for the particular breeding goal or breeding trait that you're interested in, and the other would be national average or somewhere below. And you can phenotype those animals and just use ordinary statistical approaches to estimate what is the expected differences. The main advantage is it's a vital insurance policy. We don't fully know what any of our breeding goals are doing to traits that we're not currently measuring. And this is the same as taking out an insurance policy in your car. You just want to be sure something bad's not going to happen. The disadvantage is the cost associated with it. It's costly to do large-scale phenotyping, but it's also very, very costly to do controlled experiments, especially well-powered controlled experiments. And you need to be very cognizant of the fact that they must be well-powered. You need to avoid the type 2 errors. You need to avoid a conclusion that says there's no significant difference in environmental load between genetically elite and not genetically elite animals, if there wasn't enough power to actually pick that up in the first place. So in conclusion, phenotypes, they have been, they are, and they will continue to be one of the most important aspects of a successful breeding program and a profitable production system. We have a lot done, and we have a lot more to do. Thank you.